Today's lesson is continuing the discussion about accuracy, reliability, and error that we were having last week. So what we're going to do is we're going to take all that sort of theory that we talked about and put it into an actual question. Where do we pick out on things about accuracy? Where do we pick out about things about reliability, and how do we see error? Because it's not just about learning they exist, it's about learning when you're going through your paper, how do you find these things? So this paper is from winter 2015, so it's before your syllabus change, but we are still good with the content of this one. Okay, so we are looking here at a prac, and it's a description one. So we've got to work through everything, and we've got to pull it apart as we go. So they tell us that during respiration, okay, we know what respiration is, a chemical is produced that causes the indicator methylene blue to change from blue to colorless. Okay, so we didn't necessarily come across this before, but they tell us that we've got an indicator and we've got a color change. And we've done things with color change before. We know all about color changes when we're testing for biological molecules, for example. So now they tell us that some students investigated the effect of temperature on the rate of respiration in yeast using yeast and methylene blue. So methylene blue we've linked to our color change. Respiration is what's causing color change. So let's just pull this apart a little bit more. Effect of temperature. Okay, so this means that temperature must be our independent variable. Okay, remember, our independent variable is the one that belongs on our horizontal axis. It's the one that we are changing on purpose to actually see what effect it has. Okay, so independent variable is temperature in this case. Rate of respiration. This is something that we're going to be measuring using this color change from methylene blue. If we're going to measure it, this must be our dependent variable. So in a couple of weeks' time, we're going to look more in detail about pulling out different types of variable and what we do with once we've got them. Okay, for now we know we've dissected to say this is our independent, this is our dependent. Dependent is the one we're measuring, independent is the one we're setting. Okay, so they then tell us that a ruler was used to measure 2 centimeters from the top of 3 test tubes, and a line was drawn on each test tube. Well, measure 2 centimeters, that's quite nice and accurate. They didn't just say, put it in until. Okay, they told us it's exactly 2 centimeters, so we must draw this line. Okay, yeast suspension with glucose was poured into each test tube until it reached the line drawn on the test tube. Okay, but now we've got to think a bit, okay? Remember when we put liquids into a test tube, or even when we're looking at liquids in a thermometer, we have this funky thing called the meniscus, where it slides up, okay? Now, when they say until it reached the line drawn on the test tube, at which point are you looking at it? Okay, are you looking down at it, up at it, straight at it? Are you looking properly at the meniscus? So here, it's a little bit potentially dodgy. So we're going to put a little note here. Did they measure the meniscus properly? That's something that could cause a problem. Right, 3 beakers were labeled cold, warm, and hot. Okay, so here we don't have as much accuracy. We measured 2 centimeters, that is accurate. Okay, but we're labeling cold, warm, and hot. So we're not so sure that this isn't a bit general. Okay, 1 test tube was placed in each container. That's cool. We have 3 containers and we have 3 test tubes. Okay, we're happy with that. A mixture of ice and water was placed into the beaker labeled cold, tap water at room temperature into the beaker labeled warm, and hot water from a tap into the beaker labeled hot. Okay, this is not accurate at all. Okay, so we have inaccurate temperatures. Maybe it's a cold day. Maybe room temperature is a bit icy today. Okay, we have inaccurate temperatures. We also just have a beaker. So think of it, you just have a beaker here, right? Beakers are made of glass. There's no insulation. This isn't like a travel mug that's going to keep your drink hot. This is just a glass. So you know already that the temperature is going to change during the course of the experiment. So the longer we run our experiment for, the more of a temperature change we're going to get. And that's a problem. Remember, if we want our results to be accurate, we need to have standardized controls. We need to keep things the same. So if we wanted to keep temperature the same, we wouldn't want temperature to be changing over the time of our experiment. They then tell us that thermometers used to measure the temperature in each beaker. That's okay, that's straightforward. Test tubes were left for 5 minutes. And then 1 centimeters cubed of methylene blue was added to each test tube. Left for 5 minutes. That's quite an important instruction, because remember, we've got yeast suspension in these test tubes, but the yeast suspension is all at room temperature. So we're giving the yeast suspension time to adjust to the temperature of the beaker. So the cold one's going to get cold, the hot one's going to get hotter, the warm one is going to be still at room temperature. So leaving for 5 minutes, get to temperature. But we're assuming, because the next thing is that a glass rod was used to stir the mixture. Oh, sorry. The 1 centimeters cubed of methylene blue was added and a glass rod was used to stir the mixture. We never check the temperature of that yeast solution. We don't know that the yeast solution actually in that 5 minutes got to the temperature that we've recorded in each of our beakers. So there's a lot of room for problems here. Okay, a glass rod was used to stir so that the methylene blue spread evenly. This is a good thing. Okay, it's not one of those randomly shake. Okay, when we shake, we don't know if we're shaking the same for each test tube. We don't know if person A, B, and C are all shaking the same. So, but stirring with the glass rod, that's now a good thing. That's an accurate thing. So, a stopper, technical name a bung, was placed in each test tube and a timer started. Here we have a problem. We've got to place a stopper and start the timer. Okay, now you've only got 2 hands. One can do a stopper and one can do a timer, but you've got 3 test tubes. Okay, so here is a potential problem. Okay, so that could affect the accuracy and the reliability of our results. It could introduce an error. The time for the methylene blue to become colorless was recorded. This was trial 1. This is what we're measuring. This is our measurement. But is my colorless the same as your colorless? Okay, I might think, oh, that's colorless, and you think, ah, that's still got a bit of blue in it. So it's another one of those things where you and I might be different. So it's not as precise as it could be. We're not as necessarily reliable as we could be. What is colorless? However, they then say that the same procedure for trial 1 was repeated twice to give results for trial 2 and trial 3. Repeated. Brilliant. Whenever we repeat, we improve reliability. Because when we have repetitions, we can calculate an average, and that average then gives us a better idea of what it is. So if our experiment is reliable, we're going to get similar answers for each one. Then we know, okay, we're good. Okay, despite these inaccuracies in our method, we still have reliable results coming out of it. So despite of our errors and our problems, we can get a reliable result. The more repetitions, the better. Right, so what happens next? Figure 1.1 shows the temperature of the water in each beaker and the times in minutes and seconds for each test tube to become colorless. So let's find figure 1.1. Here it is. So figure 1.1. Now, remember, minutes and seconds is always an issue in our life. When it comes to these sorts of things, we don't want minutes and seconds, we always want seconds. So we're going to have to do some conversions here. We also have to read some temperatures because they're asking us to prepare a table to record the results of the investigation shown in figure 1. Read the temperature for each beaker and the times taken for the methylene blue to become colorless at each temperature. In your table, record the temperature in degrees Celsius. Record the times. They're really nice to us in this one. They tell us in seconds. Now remember, we've already identified when we were reading that initial SPLAB that the temperature here is our independent variable. And our times are our dependent variables. So that also helps us when we come to planning our table. We always want to take a moment with a pencil to plan a table first. And we think, okay, independent always goes in the far left column. So we have temperature, and we know we're going to put units of degrees C. Then we're going to be recording the times. Time, we're going to measure it in seconds. But then we've got to think nicely, hold on, we've got 3 stop clocks for each of these temperatures. We have 3 trials. So we need to split time into 3 for trial 1, 2, and 3. Then we're going to have 3 temperatures, and we're going to put our results in. Okay, we're going to have boxes all the way around the outside. So now we know what it is. We can make one pretty table, but we're going to need some information to put in that pretty table. So let's have a look at our thermometers. Let us zoom in so that we can see them nicely. So this first thermometer, you can see we have a 0, and then we have little lines, and then we get to a 10. So there's our 0 line, there's our 10 line, and we're going up 1, 2, 3, 4, 5 divisions, which means that each division on this thermometer is 2 degrees C. So where is our temperature reading? It's here. So this is 4 degrees C. If we look at our second thermometer, there's our long 20 line, and we're one little line above. So we are at 22. And our last thermometer, there's our 50 line. We're one little line above, which means on this thermometer, we're at 52. Okay, so then we also have to deal with these stopwatches because we want time in seconds. That means we can compare. It doesn't matter if it's under a minute or over a minute, we can still compare it. So to get our times in seconds, we need to convert. So 2 times 60, and then plus 10. Gives us that 2 and that 10. So that's 120 plus 10 gives us 130. 158, well, that's 1 times 60, so 60 plus 58. That gives us 118. And here again, we have 2 times 60 plus 7, which gives us 127. These ones we're happy with, 59, 53, 57. They're all in seconds only. And this one is 35, 39, 32. Again, all in seconds. So it was just that cold beaker that we had to convert. So now we're ready, we've got all our numbers, and we've got our plan for our table. So we're going to draw our table. So this now is something that our examiner is going to mark. So we're going to be very good students. We draw our table outlines with a ruler and a pencil. And you can pretend I'm using a ruler because I haven't figured out how to do straight lines here yet. Okay, we know we want 4 columns. Wowzers. Your tables will be a lot neater than this. Okay, we know we want the top, and then we will realize that we were a bit silly because these 3 don't go all the way to the top. Because remember, we wanted to have that space to write time across the top because all of them are measurements of time. Okay, then we need our 3 different temperatures. So we have temperature, in degrees C. We have time in seconds. We have trial 1, trial 2, trial 3. Our thermometers went 4, 22, 52. So 4, 22, 52. And we do not put any units in our table. Remember, units only in the top, in the headings. So at 4 degrees C, we went 130, 118, 127, 130, 118, 127, because it's all in seconds. At 22 degrees C, we went 59, 53, 57. 59, 53, 57. And at 52 degrees C, we went 35, 39, 32. 35, 39, 32. So we've got all of our trials in. Okay, so we've done our table. We carry on with our question. State a reason why the students took 3 readings for each temperature. And because we took that time to go through the intro, we already know the answer to this one. Because we made a note that that was fantastic that they're taking the 3 readings, because they're improving the reliability of their results. So the reason why they did this, to improve the reliability of the results of the experiment. Then they actually tell us, calculate the average time for the methylene blue to become colorless at each temperature. Show your working and record your results in table 1.1. Give your answer to the nearest whole number. Now, there's a lot of instructions there. So it's important that you take the time and you work through each of those instructions. So we're calculating the average time, we're showing our working, and we're giving answers to a nearest whole number. So we need our temperatures again. So our temperatures were 4, 22, and 52. They put the degrees C at the top like good students, so we don't put the degrees C in here. Our working, to get an average, you've got to add everything and divide by the number of things. So I've got them written down here separately so we don't have to go up and down. 130 plus 118 plus 127 divided by 3. 59 plus 53 plus 57 divided by 3. And 35 plus 39 plus 32 divided by 3. And the thing is that you can plug this into your calculator one time and get your answer. And our answers are 125, 56, and 35. Now, 125 what? Elephants. 56 giraffes and 35 chickens. I don't think so. These are seconds. So I would then add in here average time, seconds. You could write 125S, 56S, 35S, but that is a little bit dodgy because we've learned very much that we shouldn't have our units in our table. So rather adapt their space there and say you should put the proper heading here. Okay, what they want to do is not give you a hint for previous questions, but you know that this is how it should be. Headings in the table have the units, not the cells. Okay, so now we carry on with the question. State one conclusion about the effect of temperature on the rate of respiration in yeast. So, the effect of temperature, our independent variable, on the rate of respiration. So what happens? We know that we looked at temperature increasing. And what happened when we increased the temperature is that we turned color faster. So if we're turning color faster, it means we have more respiration that's happening. So as temperature increases, rate of respiration in yeast increases. That's it. Straightforward, plain and simple. As temperature increases, rate of respiration increases. Remember that here we're measuring time to go colorless. Okay? Time to go colorless is inversely to rate of respiration because more respiration means less color. Okay, right, so the method of timing how long it takes for the methylene blue in the 3 test tubes to become colorless is a source of error. Suggest why. Well, Mr. Examiner, we're ready for you because we already thought about that when we did our introduction. We had 2 different things. One was that it's difficult to do all of them together because we've only got 2 hands. The other one was, is my colorless the same as your colorless? So it's only 1 mark. You need 1 reason. So here is your choice. You can say it's difficult to watch all test tubes simultaneously. Or you could say that it is difficult to judge the endpoint. Now the next part is, see this is C part 1 and C part 2, it means they're linked together. Okay, describe how you could improve the method to reduce this source of error. So whichever one you pick for C part 1, your method improvement must relate directly to that error. So if we pick the first one, it's difficult to watch all test tubes simultaneously, our fix would be do them one at a time. If we pick the second one, it's difficult to judge the endpoint, we could use a colorimeter to judge the endpoint. Okay, but it is absolutely crucial that whichever one you pick for the error, then you pick the relevant fix. You can't give one error and then give a different fix. Okay, they have to be related to each other. And that's the same when you're talking about any kind of error in any fact, they must be related. Okay, so moving swiftly on. We have a graph. Okay, so I've also just been asked a question relating to this, is what is a colorimeter? That is a very fancy machine that can measure color really accurately. So whenever you're looking for a color change or an endpoint of a color, reaction, then that is your fancy piece of equipment that you dream you had in your lab. So moving on to the graph, we've got to now look at another investigation. We've got to put some results into a table, plot a graph, and then describe and explain from the graph. So we only have 3 minutes left because we've been focusing on this accuracy, reliability, and error. So we're going to skip these ones for now. We shall potentially come back to those in another live lesson. We take a bit more detail again, looking at the graph and interpreting graphs, because it's a really important skill as well. So for now, you've now had 2 sessions on accuracy and reliability and error. So hopefully they make a bit more sense and you can distinguish between the two. Remember that your accuracy is am I going to get the same thing that I'm supposed to get? Reliability, do I get the same thing every time? And error are potential problems that can be introduced into the experiment because of the way we do it. So whenever you're looking for accuracy, reliability, and error, it's always coming out of this initial description of the experiment. That's where you got to keep your eyes open. You've got to think through what's going on and pick out what are things that are going to be problems potentially later on. Then by the time you get to the questions, you've got a thorough understanding of your experiment and you're ready just to answer them.